Hi, everyone. Hope you're going well. We're just going to wait a minute or two for some more people to join, and then we'll get going. All right, I might get started. Welcome to the Building Consensus for Marine Microplastic Monitoring session at the Ocean Best Practices Workshop. My name is Nina Wooten. I'm a marine researcher down here at the University of Adelaide in South Australia. We're excited to have a great global spread of presenters here today to discuss what is happening in the different parts of the world in the microplastic space. Before I get too far in, I would just like to acknowledge that I am presenting today from the beautiful Ghana country, also known as Adelaide. I would like to pay respect to the First Nation peoples across Australia and offer my respect to their culture, caring for country, and elders, both past and present. I know that we have people here today from all over the world, from different countries and regions, and we are so excited to have you all here today. I've seen a lot of people are already saying hello, but if you want to pop your name and where you're from in the chat box, that would be really handy, just to see where everyone's coming from. But it's great to see such a broad and global spread of people. Just a bit of housekeeping before we get too far in. Of course, I think we're all used to the Zoom era now, keeping your microphone on mute. That will be really handy. And we've got a pretty tightly packed schedule. So we're going to answer the questions all together at the end in question time, where we'll have a bit of a panel session. If you can just put your questions in the chat, there's a Q&A box, I believe. If you pop them in there as we go, that would be really handy. And then I'll report them back in to the different panelists and people as we go along to see what people have questions for. So the workshop agenda, I'll just quickly run through who we have presenting today. So we've got a really great geographical spread of presenters. Shout out especially to Jen for getting up at 4:30 a.m. in Canada. You must really love microplastic standardization. There are 6 talks that we have prepared for you. So first up, there'll be myself and my colleague Patrick. And then we're going to hear from Dr. Denise Hardesty, who will provide her expertise from an Australian and an East Asia perspective. After that, we'll have Dr. Amy Lusher, who will give us an update on the technical readiness in marine microplastic monitoring, focusing on Europe and the Arctic. And then we're going to have Professor Ted Henry, who will give his expertise from a UK perspective. And then we'll have Dr. Sheree Motti from the Australian Institute of Marine Science, who will speak about data reporting for environmental management and societal change. And then last but not least, Professor Jen Provencher, who, as I already mentioned, has got up very early for us, but she will be giving us some insight into what is happening in Northern America and harmonization in the field and standardization in the lab. So then following on from these presentations, we're going to open up the floor for some questions. Hopefully we'll have some exciting discussions as part of that. We're hoping to finish up at 10 o'clock UTC, transfer that into whatever time zone you're in. But we do have a bit of leeway to go a little bit over. So if our discussion and question time is going well, we'll see how we go, but we have to be finished by 10:30 UTC. All right, I think that's all the housekeeping and agenda information. I'm going to hand over to my colleague, Dr. Patrick Rees Santos now, who will start our presentation about standardizing microplastic sampling from an Australian perspective. Patrick, I'll just head over to the next slide and mute myself so you can have the floor.
Thank you, Nina. Thanks for that introduction, and a warm welcome to everyone in our session. I'm really thrilled to be here today, excited to give you a quick overview of why we are here today and the pathways that led to all the current efforts we are developing in terms of standardized methods for microplastic sampling here in Australia. But first, really, we really want to acknowledge that this was a truly massive collaborative effort, bringing together over 40 experts from 21 different research, industry, and government institutions. And so to us, it's really important to emphasize how important this collaborative, cohesive community approach was for the successful development of the standards and the current implementation process we have ongoing for microplastic standards. So really a big thank you and a big shout out to everyone. So we know and mostly agree that standards, best practices, and method harmonization are incredibly important. And they open all these possibilities from enhanced data comparability that leads to greater trust in data sets, to allowing for larger collaborations, for improved systems of interoperability. And of course, I think quite importantly also to optimize our time and our resources. But while we often agree we should have a standard, no one really wants to use someone else's for several reasons. And that could be just like we think about that idea I have there of a toothbrush. But I also like to think of standards as cooking recipes, because as cooks, we all bring our unique flair. But standardizing or following the recipe really ensures consistency, meaning that your famous dish really tastes the same every time, no matter who's cooking. And the same really applies to scientists and our research. So how did we end up building these best practices for microplastic in Australia? What's the story here? This really all began as part of a small regional project that combined a review for microplastic data at the time, online surveys, and a workshop with industry, researchers, NGOs, and government. And this was driven by local government agencies wanting to understand what were the gaps and the research needs in terms of the microplastic space to actually allow translating that microplastic research and information into policy and action. They wanted to fill that gap. And so following a large consultation and discussion period, really the overwhelming response was summarized in these three main priorities I can show here. So method harmonization, understanding present source and pathways, but also risks in terms of effects. But really method harmonization was a clear leader. And I believe this won't come as a surprise to many people in this room, but this horrendogram I'm showing next shows incredible diversity of methods that we really identified from the review section of our initial work. And this really is for Australia alone. Now, all this really limited our ability to synthesize and compare the data that was available at the time. Also because not only the methods varied, but the description or the reporting of those methods and the results missed critical details that really limited study comparison. And for any example, they really limited synthesis in terms, for instance, of spatial and temporal trends of microplastics. So this is where an idea of developing standards started to take shape. Australia's National Environmental Science Program, championed by Rachel Przeslawski at the time, decided that there was such an overwhelming evidence and institutional support that microplastic standards were needed, that they added it to a list of developing standard operating procedures they have been developing over the last few years. And so now to really dive deep into the development of these standards and what we've been doing, I'll hand it back to you, Nina, who will talk us through the process in a bit more detail.
Cool. Thanks, Patrick. Great to see an overview of how this project that we've been doing all started. So I'm going to just touch briefly now on the kind of framework that we followed to get an ocean best practice. And we are in an ocean best practices workshop. So I'm sure people may have seen this image before. But in short, this first step of the confirming the need, at least in an Australian context, and I know we're going to hear some really great stuff that's happening internationally as well. But we definitely decided we needed this this set of standard ocean best practices for microplastic sampling through what Patrick was talking about. So the project we talked about before. So following this, we already had some people that we knew in the space, but we obviously knew who the people working in Australia were in the microplastic space, and we formed this working group. So that was the team of 40 researchers that Patrick mentioned at the beginning of his talk. We became this sort of working group that included researchers from academia, government, industry. We had NGOs there, and we were really happy with this sort of broad, I guess, acceptability and group of people that came together to help with this problem. And then we spent about 8 months working on developing the content for in a kind of Strawman draft. So this is this section here, the developing content section, and we focused on what the scope was going to be. This took a few iterations, and there are a lot of Google Docs floating around, and anyone that was part of our working group probably got very annoyed by my emails, but we finally got there. We had a final draft and it had international review, and we're really excited by that. And then now we're finally at this point here where we're releasing the online documents. And yeah, we'll be a bit more in touch on that a bit later on. I've got a link to show if anyone would like to see them. But I thought next I would just step through like a super broad overview of how these documents fit together. So the first discussion that we had as a working group, and it was for quite a while that we had this, was which matrix we were going to attack. So I had this very naive idea that we might be able to put it all into one sort of manual, but we ended up effectively having 4 because each matrix, obviously, although they do have a lot of similarities, and you can see this here by the overview that we have at the start with the glossary and the intro and scope, goals, context, etc. Of course, we all know that there's different particularly different collection methods, but also the processing does differ a lot depending on the matrix. We weren't going to have air initially, but then it got added on as there was a lot of interest in that space and not a lot done in that space yet. So hopefully this one here will come in a lot of use. And then the second broader discussion was to do with, I guess, the level of detail or description that we were going to provide. So I'm sure many people in this room understand that any one of these manuals could singularly be hundreds of pages long. But we wanted to make sure that we had this sort of sweet spot where the manuals had enough information to have harmonization and reproducibility, but also not so much that it was tailored to specific labs or equipment. And as a group, as an add-on to this, we decided that having a checklist at the end was really, really important. So not just quality assurance and quality criteria, but also a strict list of the criteria that needed to be reported to allow you to have harmonization. And that's can be found at the end of the document as well. So just really briefly, this is super high level, but we focused on, there's an image actually out of the field manual, and we focused on different levels. So we had some levels of higher quantification, and this was to give flexibility to allow NGOs and citizen science groups, and we had a few people from both of these in our working group. We wanted them to be able to collect data on abundance and presence as well. And then we went all the way down, of course, to the more detailed level of chemical characterization with polymer type, and there's a whole range of different ways you can ID polymer type, which I'm sure lots of people know about as well. So that's a bit of an overview of the kind of level of depth that we went to. And then these documents now are available on the Marine Sampling Field Manual site. So the ultimate aim was, as a new researcher to the field, that you could pick this field manual up, whether you're working in academia, an NGO, and then you could use them as a guide to produce accurate and comparable data. And this came from, I guess, it stemmed from the fact that when I first started my PhD back in 2017, I didn't really know. I was sampling microplastics in seafood, and I got very lost in the methodology, and there wasn't a clear method then. And I know there's a lot that has come out in the meantime, and we definitely align with those, which I'll touch on in a minute. But I just wanted, yeah, that was kind of the reason why I felt particularly passionate about this. So as you can see here, they sit amongst a suite of other marine sampling field manuals, and these are all have come from mostly Australian researchers. But a lot of them now have almost become this bread and butter for sampling methods globally. Particularly the beta remote underwater video one. I'll highlight that there. That's a really great document, and I know it's used by a lot of research groups all over the world. So yeah, feel free to scan the QR code if you want to have a look, or I'm sure that we can send it around at some point following. And yeah, I just wanted to highlight this work is not in isolation. This links to the other field manuals that we looked at before, including the ones on how to design your survey. But in particular, we really wanted to make sure that these build and link to global efforts that already exist to standardize methods for microplastics worldwide. So that includes AMAP, the Arctic Monitoring and Assessment Program, which I know both Amy and Jen have lots to do with, and I'm talking about later on, but also GSAMP, and of course NOAA as well. So there's even a whole section in the field manuals that sort of puts this work in the context of what's been happening globally, so we don't do things in isolation. So there were a few challenges, of course, and there were some within our group, but we were really lucky that we already had a group that we'd kind of built upon and we could build momentum from. But I guess that there were a lot of methods that everyone was already using, and formulating a protocol that like combined everyone's together was not an easy task, but we got there in the end. And yeah, we wanted to make sure that these protocols had broad application without losing the detail. So I guess the next steps for us is we've released our documents, and soon these will be uploaded onto the Ocean Best Practice repository. And then, of course, and this is why this workshop today is so great, we want to continue to build on this global momentum and take next step forward to make sure that globally we're all working together so we can make, I guess, have a good quantification of microplastics and can compare what's happening all over the world. So that's the end of me talking, although I will be talking a lot more as I share the next presenters and let you know who's coming up next. But I'll stop sharing now. So I think now we have Denise. So Denise, if you wanted to share your screen, and you can introduce yourself and get going. I'm going to pause my audio.
Super. I'm just hopeful that people can hear me and see my screen, which
yeah,
trying to give it full screen view. Super. Well, thank you. All good? Okay. Thank you so much, everyone, for joining. It was really heartening to see people are coming in from all over the world. So good morning, good afternoon, good evening, depending upon where you're joining this working group on today. I am dialing in from the land of the Murwinna people in Palawan, which is in Tasmania, which is in the very small little island part of this map. And I would also like to just honor and pay my respects to the elders past, present, and emerging, as well as to honor and acknowledge that this is country or land never ceded, and also to acknowledge their continued custodianship to and connection to land and sea country. So I work for CSIRO, which is Australia's federal research organization, and I've been leading a suite of projects looking at plastic pollution for the last 15 plus years. And our team focuses on understanding what and where and why and when do different types of plastic and other debris types enter the environment. And this is sort of broad scale. So understanding what relates to that: people, population density, road networks, infrastructure, etc. Then we have a body of work trying to understand, how does our waste move through the environment? You know, is it moving along road networks, along rivers, creeks, and streams? Is it by dumping, etc.? We've also done a fair bit of work looking at the impacts, and I'm going to say plastics for all of these things, because we look not only at microplastics, but also at larger debris items. And so I will talk to that a little more specifically, because here today and tonight we're really focusing on microplastics. But we've also done a body of work to look at the impacts on wildlife, seabirds, marine mammals, turtles, as well as looking at some of the impacts potentially, on people and economies and wellness and well-being. And then the fourth really large body of work that we've done is actually evaluating some of the policy or management responses to try to understand, how do you implement policy and what's going to be more effective in different contexts? Is it outreach and education or legislation, and understanding that in a variety of different contexts? So I say that as framing. As Nina mentioned earlier, many of us have been working around the world and on this topic for quite some time. And we've really been trying to understand how our waste and debris moves from inland areas out into the marine and coastal environment. And so the components that are really relevant for microplastics per se are focused so much on what's happening in the floating ocean, as well as along land and rivers and creeks and streams. And we use a statistically designed, robust sampling method to really focus on that interoperability or comparability that you've heard, which will be a refrain throughout this session, I'm sure, from all of us. So we're really trying to focus on making sure that we can make comparisons at different scales in different countries around the world. And making sure that we can do something that, with a bit of training and volunteer participants, this work can be analyzed, be robust, be rigorous, be repeatable. And so the process that we take is using a stratified random sampling. We conduct a series of transects, and we're focusing on those items of debris that are larger microplastics. So those things that are visible to humans at standing human height, or that we can see and observe from sea surface trawl samples. Then the data is all collected and recorded. And again, I just want to highlight the importance and relevance of using the categories that are commonly used, because that allows us to make comparisons among different strata, among different sampling procedures and processes that are taking place in other parts of the world. And so again, the part that's really particularly focused on the smaller items are those things that we see in floating in the global ocean. And our focus has traditionally, or more typically, been near land, so in the near shore environment. I will say, however, that in nearly every surface trawl sample that we do, we also get debris items that are far larger than microplastics. So we, again, don't limit solely to focusing on the small items. Just to give a little bit of an example, this is just 5 countries where we've been recently working. As we talk about taking a regional approach, we've been working across Cambodia, Philippines, Thailand, Vietnam, Malaysia, as well as another 15 or 20 countries around the world. And here just shows the representation of what we may do within a geographic expanse, so that you're, again, while the landscapes are different and the needs may be different, by using a consistent approach, it allows us to make comparisons within and among regions of the world. And that becomes really important, whether we're talking about different taxa, different geographies, different strata, and making sure that we really collect information in that consistent manner. And to me, I suppose there's just a few things I wanted to highlight that, as was mentioned earlier by Patrick, we all think it's important to brush our teeth, but nobody wants to use somebody else's toothbrush. Really think about what your questions are, so that your approach allows you to address the questions. And so the one piece of recommendation or advice that I would give is to please try to align your data collection with established methods, and then you can add or adopt as needed, so that, the way I was thinking of it is, don't reinvent the wheel. You may need to realign a little bit. You may want to add a particular field or code, but allowing and presenting and sharing data in a way that others can also use that information to expand their analyses will allow us to have much better outcomes overall. I would also really encourage, as Nina pointed out previously, the value and importance of running controls and tests. So positive controls, environmental controls, negative controls, particularly as we talk to the smaller sized particles, and particularly if people are interested in leachates and other things that may be coming out of these chemicals, it becomes really, really important to run those types of controls and checks. And also to acknowledge that every method has its flaws and is not perfect, but I would argue that perfection is the enemy of done. And so I would encourage us to just take note of the limitations, acknowledge what's going on with the data set that you have and the approach that you're taking, and really try to encourage people to align and to acknowledge with what you're doing so that we can build that global body of work so that we can see methods be adopted and adapted in a variety of different contexts. And I will leave it there. Thank you so much.
Thank you, Denise. That was awesome. I always love what people are seeing, what people are doing, people that I already know what they're doing, because I don't often get to see it in this kind of context. So that was really cool. I might get you to stop sharing now and move on to Amy. We're still on time, so this is looking positive. Next up, we have Amy Lusher. Do you want to share your screen, Amy? I'll stop talking. Perfect.
Sorry, I couldn't find the unmute button. There we go. Hi, everyone. I'm Amy Lusher. I'm a senior researcher at the Norwegian Institute for Water Research. And I'm going to talk to you today a little bit about some of the work we've been doing both in Europe and in the Arctic about identifying the technical readiness of our methodology that we use for microplastic assessment and monitoring. We've already heard a bit about this already this morning. There are so many different approaches that are being used to analyze microplastics in the environment. And this can sometimes hamper the setup and the design of large-scale monitoring programs. We see differences between individual research teams, countries and regions, and this will have consequences for monitoring, risk assessment and legislation. Since the field is still evolving and we will continue to see new or improved analytical methods being provided on a regular basis, we need some tools or a way of helping identify which methods or approaches are appropriate for our research or our monitoring. So this can be from a researcher perspective who wants to start microplastic research, like how do you handle all of the literature that exists in the world of microplastics, but also for monitoring campaigns for an individual countries or regions want to start monitoring plastics. Sometimes they just don't know where to look or where to start to identify which methods work. As Denise and has already mentioned, and Patrick as well, that sometimes individuals like their methods more than others, and we need to find levels of compromise where we're finding methods that work the best for us. So since we have different research questions, they will have different approaches. I won't go into detail too much on the figure here, but this is just trying to explain how monitoring methods or methods are integrated into monitoring and assessment. And when we first develop new methods, we have to test them for their feasibility and their validation before they get recommended as standardized approaches and picked up within monitoring. And then we always go back over them again and see, are these methods doing what we want them to do? So when we want to develop methods that are fit for research or monitoring, it needs to be a key step at the very beginning of any microplastic pollution control and management. We've heard the word standardization and harmonization already today, and these are both important steps for us when it comes to evaluate and optimize our methods and approaches for microplastic pollution monitoring. I argue that harmonization is more important than standardization, mostly because nobody's going to be able to use exactly the same methods. So there needs to be some level of flexibility in what we're doing, especially because we need to be able to adapt to different scientific, logistical, environment, and ethical constraints, which varies around the world. But it's most important that we're harmonizing what we're doing from the beginning, from survey design right the way through to data reporting. Because if we do make changes in methods, we have to consider what the consequences will be for our data reporting at the end. So every change that we might make to a agreed upon method could have consequences to our overall reporting. During the EuroQChan project, which ran from 2020 to 2023, we were looking at trying to find a way to help us make decisions on our methods. So we decided to use a technical readiness scale, which is developed from NASA, which was used for space technology and readiness in space technology. So we divided the analytical chain into digestible steps, and then within each one of those orange arrows on the screen, that was broken down into more fine details. But in short, the TRL scale runs from 1 to 9, where 1 is the basic principles of ideas about a method, whereas TRL 9 is where you have a standard protocol that's enforced and it's widely applied and used for monitoring operations. So we use this scale to look at all the available methods for microplastics and plastics monitoring. And I can't go into the details on all of the outcomes, but if you want the details, I can send you links to all of the reports. But the main outcomes that we had were that only a few matrices and size classes had a high level TRLs in all steps of the assessment. So that's from data survey design through to data reporting. And macroplastics is one of the examples here, because macroplastics monitoring started much earlier than microplastics monitoring. So we've had more time to align our methodologies and start developing similar approaches. When monitoring programs are being set up for microplastics, we saw that the biggest challenge was representative sampling. So there was a challenge on the amount of samples or volume of samples that should be collected. But then there was also the additional challenge of how to assess microplastics in complex matrices, because these methods were very, very varied between different individuals and research teams. We identified that processing protocols and all of the different variations that exist that were required to obtain scientific data tended to be related to projects with short timeframes. So usually over one to 2 years, and they were limited to a certain number of samples. So they weren't often suitable for large-scale monitoring programs, which are driven by cost and reproducibility. So before we could integrate such processes into large monitoring programs, we identified that rigorous quality control was necessary through intercalibration testing, for example. And at the moment, no sample preparation protocols for microplastic particles, this is specifically less than 1 millimeter in size, have successfully passed any rigorous laboratory testing experiment. So there are lots of interlab comparison exercises ongoing, but they don't necessarily have positive outcomes at this moment. But hopefully that will be improved soon. So if we take this information and put it into the Arctic context, we see the Arctic's not free of microplastic contamination, and most of the efforts to track microplastics have been uncoordinated. As an example, my own piece of research here, we tried to understand what was going on in relating to walruses and their exposure to microplastics. So we looked in the water, we looked in the sediments, and we looked in walrus poo, but we didn't actually look in the prey species that the walruses were consuming. So these sporadic investigations often give us an incomplete picture of the spread of microplastic contamination. And again, wide spectrum of methods is being used. So through AMAP and through PAIM, there has been some understanding of what level of data that we have, and mostly it's limited. That's within the freshwater and marine compartments, comparing water to sediments, and also looking at sources inshore and offshore. So we had the motivation to develop a pan-Arctic monitoring program within the AMAP working groups. And if this was mostly linked to focusing on efforts to build an understanding of microplastic pollution from the local scale to the full pan-Arctic. We wanted to understand how plastics and microplastics got there and what the impacts it could cause. And this required, in order for us to do this, we wanted to have a good understanding of the source contribution so that we could implement pollution avoidance and mitigation measures. In order for us to develop in the monitoring strategy, we knew we needed to consider the policy question that was being addressed, as well as the resources that we had available to carry it out. So it should be cost effective to ensure that monitoring could be maintained. We needed to prioritize to address significant risks and associated indicators, and encourage cooperation. This could be building on existing monitoring activities, but we also wanted to favor innovative and opportunistic approaches to collect data. Most importantly, the methods had to be adapted to the Arctic ecosystems. So, for example, we had to consider local site conditions, whether that's highly polluted with carbon particles, which can affect microplastic analysis, the proximity to anthropogenic activities, and also the presence of fauna in our sampling locations. So it's really important to consider multi-matrix monitoring rather than just looking at a single matrix. So the joint sediment and water approach can be carried out in the same sampling campaign. They can provide complementary but not overlapping information, and it provides the most complete picture of plastic pollution. So looking at water, we can look for track rapid fluctuations in an environment, but we can also look at the sediment for spatially and temporally integrated signals. But we do appreciate that most of the data in the Arctic is still biased towards the marine, so we need to increase freshwater and terrestrial samples. When we're monitoring the level of change, we need to look for representative locations. This requires pre-existing knowledge and initial screenings. We need to assess variability to understand the level of statistical power, like Denise described. And we also have to consider cost and logistics, which is probably the most important. And ancillary data and contamination are another very important step that we have to undertake for Arctic research, because we can't avoid plastic in many cases. So just to wrap up some of our key messages here, a harmonized and coordinated effort is needed to gather information in Europe and the Pan-Arctic. And this, to understand a full picture of microplastic pollution in an area, requires knowledge of aquatic and sedimentary systems, as well as the links between them. And multi-matrix sampling is advantageous for building a full picture of environmental microplastic contamination. And this does allow us to identify priority regions for focused mitigation efforts. And we should also make sure that the monitoring that is performed aligns with the priorities of local or regional Arctic communities. And I'll stop there. Thank you.
Amazing. Thank you, Amy. That was really good. Wow, I feel like I learned so much in that 10 minutes. If you want to stop sharing, we might go on now to Professor Ted Henry. So Ted is from Heriot-Watt University, and Ted is going to be presenting on the substances associated with microplastic particles and their potential to cause toxicity. I'll let you share, Ted. That's good.
Okay, I should be sharing, hopefully, if that's working.
It might be going presenter mode. Is it presenter mode yet?
There we go. Is that both sharing and you can hear me?
Yeah, that is perfect. You're good to go.
Marvelous. Okay, thank you. Okay. So first of all, thank you very much, Nina, for the opportunity to present here. Just to introduce myself, I'm a professor of environmental toxicology here at Heriot-Watt University in Edinburgh, UK. I'm going to talk a bit about substances associated with microplastic particles and some of the things that we need to consider in that context as we consider some of the methods and the important aspects of that. So if we think about a plastic particle and the substances that are part of the particle or part of the plastic when it's manufactured, put that in water, and then there's some dissociation of those substances, and they then get in the water. And is that a concern for organisms that are exposed to it? We've worked on this topic to some extent, and I something coming up on my screen here that's blocking. Okay, and so here's just a quick example. So imagine PVC particles, polyvinyl chloride particles, and in some PVC formulations, lead has been used. And so we made some particles out of this, and then demonstrated that some of that lead can come out of the plastic particles. And we used our zebrafish model and a gene expression of the metallothionein gene to show that there was a relationship between the concentration of particles that the lead leaked out of the plastic particles, and you could see that became bioavailable in the organisms. So that sort of thing can happen, but notice the concentration is really quite high, and so that's something to consider in terms of the relevance of the effect of these types of substances. If we consider the sort of another scenario that is quite prominent in the environment is that the plastic particles are out in the environment that are associating with other substances that are present there. So any number of different types of toxic substances. There's been numerous analyses of plastics that have been collected from the environment, and numerous substances that have been detected. But some of the things that we need to consider in this context, and how important is this in terms of the various things that we need to consider in terms of the environmental concerns around plastics and microplastics. How much substance is associated? What is the nature of this association? Is it strongly held such that it's not going to be becoming bioavailable? So the question of bioavailability is very important. What are the scenarios and how should we investigate them? And really, are these substances of toxicological concern? So we realize that our analytical chemistry techniques are so sensitive that we can detect extremely low concentrations of substances. And therefore, there is a question, is that of toxicological concern or not? So we've looked at this in a variety of scenarios. I'm going to show some different scenarios, also using some of the work we've done with engineered nanomaterials, because there's very close similarities. And so the thing to consider in this context is the topic of sorption and how substances associate with particles. What happens in that scenario when we put a plastic particle in the environment? It becomes quite a complex assemblage of multiple things that are going on the surface of that particle. There tends to be a biofilm formed. Substances are going to associate with that biofilm. When they get in there, they're going to then be able to be adsorbed onto that surface and then absorbed into the matrix of the polymer. And so we have to appreciate that that's quite a complex interaction. There's microbes on the surface of this. You can show that these microbes can change the contaminant that's present in that association. So another thing to recognize, and as a toxicologist, we've worked quite a bit on trying to understand how do we test the toxicology of particles, whether it's an engineered nanomaterial or a microplastic in this case, and tried to come up with some standardized designs to be able to make the results comparable. And one of the key tricks is trying to make sure that your exposure is reproducible. So we've done various things to try to generate homogeneous exposure scenarios. This is an example of that, where the particles are maintained in a suspension, the organisms are kept in a way that exposure can be consistent over time and be reproducible. When we think about this topic of plastic particles in water and the sorption topic, it's really about this equilibrium reaction. So there is a plastic particle, that plastic particle in water is going to reach some equilibrium in terms of the substances that are present in the water are going to sorb to that particle, and that's going to be in some sort of equilibrium scenario. And so in the context of assessing this and what is important, one of the things that's important is trying to understand, well, what is the bioavailable fraction of this scenario? If the particle comes in association with the organism, do those substances come off, and are they bioavailable? So there's different ways of looking at that. We've used molecular techniques, gene expression as one way to assess whether some of these substances are bioavailable or not. I'll show you a couple of examples of that. So here's an example with some particles. These are actually engineered nanomaterials looking at this sorption between mercury and titanium dioxide, and using metallothionein expression. You can show that the characteristics of the particle, the surface area and size of the particle, leads to changes in the amount of material that's present in the aqueous phase. One question that, so we can certainly demonstrate and have, as numerous others have, that substances are associated with nanoparticles, plastic particles. Here's an example where we were testing whether there was association of the ethinyl estradiol, which is an estrogenic substance, and that certainly can associate with numerous types of particles, including plastic particles that we've tested, and show that association occurs. One question then comes up as well, okay, if an organism concentrates these particles and then it's ingested by, for example, a fish, does do these substances come off and lead to an exposure? Do those substances become bioavailable in that scenario? We found very limited bioavailability in that scenario. So it's certainly not all substances have been tested, but it's quite a low bioavailability that we have seen in that situation. So the question is, so can the particles enhance bioavailability under some type of situation? So if we imagine we've got some particles in the aqueous phase, they're absorbing different substances. What's the nature of the effect in organisms? Well, we've tested this scenario in numerous types of particles, and this is an example of something that can happen. So the particle agglomerate absorbs to a substance. In this case, it was mercury. And that changes the physical chemical characteristics of the particle and can enable that particle to sediment down to the bottom of the container. And so if we were then to test this scenario, if we look at the mercury concentration in the water column, the mercury concentration has decreased from the container on the left to the container on the right. It has decreased because it's sorbed to the particle and brought to the bottom of the container. But interestingly, if you have organisms that are at the bottom of the container, the question is, does that substance that is associated with the particle have increased bioavailability? We demonstrated that in this case, and in a couple of other cases, we've also seen that there's scenarios that can occur, specific situations where particle can sorb a substance in the aqueous phase, bring it down to the sediments, and enhance its bioavailability in that scenario. It turns out that if you hold the fish up in the water column, you eliminate that effect. Taking a look at some of the microplastic particles and a variety of things that we're working on just now, and trying to understand, well, what is it about certain particles that lead to the absorption of substances with their surface, and how does it enter into the matrix? We find that the different surface chemistries is important. And here is an example where we had PA, PE, and PET particles, quite different surface areas, but what we're showing in the figure here is an amphipod organism where we're looking at whether the substance becomes bioavailable or not, and if what is the difference in different particle types in terms of their effect on sorbing the substance. We find that with the same concentration, we didn't see any differences, despite quite orders of magnitude differences in the amount of surface area that was present. So there's certainly considerable differences in how substance is sorbed to the particles, and this has an effect on how much substance is present and associated with the individual particle. One of the things that comes up quite frequently that I hear people discuss is this topic of microplastics and what are the risks to organisms that ingest food that is contaminated with microplastics. And this includes humans. And you'll see certainly reports where there's concerns about, okay, are people exposed to microplastics through eating seafood, for example? And is one of the risks there something about the substance that are present with the microplastics themselves? So we've looked at this in some ways, and just approaching it from a literature standpoint. And I'm going to leave you with this thought, which is really to try to appreciate the context and what are the key things that are important. So if we take an organism like a marine mussel here, which has the potential to accumulate microplastic particles, and then humans, of course, ingest mussels. And so if you think about the contaminant lead and what we found in the environment, that microplastics can contain the highest levels that we've seen in the literature, about 0.01 micrograms of lead per milligram of plastic. So we can have an idea of, okay, how much lead is present on the plastic, and then consider, well, okay, how much microplastic exposure do you get with a seafood meal? And so in the case of mussels in this example here, the amount of microplastic exposure, 0.26 milligrams in this context, which is consistent with numerous studies in the literature. So that's how much microplastic. How much microplastic would that have? How much lead would that have if it was completely containing lead at that level of microplastic contamination? And you can see that the amounts are just extremely low. And so if you were then to extend that and consider, okay, how much lead contamination for efficient consumption advisory to occur, and you'd see that it's really orders of magnitude higher than anything that we could consider happening from the microplastic. So I'll leave you with that thought before I go to my acknowledgement slide, just to remind us about thinking that, so what are the key issues of microplastic contamination that we need to think about from a toxicology standpoint? And I think some of these associated substances may not be as important as sometimes we're led to believe. So numerous people to acknowledge. We've been working on particle toxicology and substance absorption for quite some years. And these are just some of the people that have been involved and some of the funding organizations that have supported the work over the years. Thank you very much.
Great, thanks, Ted. That was really interesting to get down to that particle and toxicology kind of perspective. A whole nother kettle of fish on standardizing microplastics contamination if we're using it in experiments. So that just, maybe that makes me feel more overwhelmed, I don't know, but awesome to hear anyway. So if you stop sharing, oh, Shree's already onto it. So now we have Dr. Shree Motti, who's from the Australian Institute of Marine Science, and Shree's going to be presenting on microplastic monitoring in Australian marine waters, operationalization and data reporting for environmental management and societal change. Shree, I'll pass over to you. I'll mute myself and you unmute yourself.
Lovely, thank you. Can you hear me all right? Perfect.
Yeah.
Thank you for the introduction there, Nina. So yeah, I'm just going to dive straight into it, and firstly do a quick acknowledgement to country. I'd like to acknowledge the traditional owners of the land and sea pretty much around the entire Australian coastline, because that's where our work is conducted, and in particular up here in Townsville, the Bindal and Walgarookaba peoples, and pay my respects to their elders past, present and emerging. So I'd just sort of like to start with, microplastics. Why should we care about them? And some of you may know, but we have what we call the 9 planetary boundaries, and currently 6 of those boundaries have the thresholds have been breached. And this is really worrying because part of the issue is the novel entities, which is associated with the chemicals that we produce and use. And within these novel entities, plastic is actually being identified as pretty much one of the main threats. And in particular, recently, microplastics are now deemed as a planetary boundary threat. And so what does this really mean? Well, we know through the production, transport of it, recycling efforts, that plastic's actually contributing to climate change and ozone depletion. We know that through its release, whether it's intentional or not, and into the environment and its persistence in the environment, that is starting to have real impact on land systems, on freshwater use, and also in atmospheric loading. And of course, they are also having, through interactions with biological, physical, and chemical systems, significant impact on our biosphere integrity and on our biogeochemical flows. And more recently, there's even evidence that ocean acidification is starting to be impacted. And so this is why we actually should care. We should be monitoring microplastics so that we can really understand what is actually being entered into the oceans, and of course, how long they're going to be persisting for. Now, I'm not going to go through this slide too much, but Amy already did a nice description on how you define your problem and why it's important to do so. And so 10 years ago, when the AIMS Microplastics Group started up, that's exactly what we set out to do, was define whether on the Great Barrier Reef there was actually a problem or not. And we focus particularly on some coastal and offshore transects from the Townsville region, shown here in green. And we found that every sample we picked up had microplastics in it, and we were really quite surprised by that. And so then what we thought was, well, how significant is this? What is the potential impact of these plastics? And so we undertook some modelling to see where the potential trajectories might take these items if they stayed in the oceans. And what we actually see is that they actually have a great capacity for being transported through our system. And so this really made us realise that we actually did need to do something about this. And so our first course of action was to actually measure what was happening. And so our action here was to develop a monitoring methodology. So we set about establishing and validating methods, testing the feasibility of these methods in an operational aspect, and of course, generating data. And all of this is designed so that we can have a better representation of the problem, a better understanding of the problem, and of course, be better informed on how we can use this information to manage plastics in the environment. I'm not going to go through the methods as such because Nina has done a fantastic job in introducing the methods, and we've worked really closely with Nina's group to bring our methods into that documentation that she spoke about. But briefly, what we focus on is having a validated workflow whereby we collect using surface tows, we apply density separation and filtration, we conduct a visual assessment, and then we do microplastics assignment. And we do all this under a strict QA/QC protocol. And so we were interested in what was happening in our backyard. And so we took our methods that we'd been developing and undertook a pilot study to do a temporal assessment at the shipwreck Yongala. So over a period of 4 years, we conducted a series of transects. And alongside that, we were also collecting water quality data and environmental parameters. And what we were able to observe when we plot out the data is that we see the baseline down here. It's sort of got a little bit of variation, but it's pretty much an even baseline, except that we had these two major outliers. And when we look back and compared at this data with the water quality parameters that we have, we could see that there was a correlation with low winds, high river discharge, and low salinity with the high plastic levels reported. And so that then made us look into what else was happening in this space at the time. And in fact, what we saw in 2017 was Cyclone Debbie occurring. And then again in 2019, we had the 1 in 100 year floods. And so we saw this significant spike in the data because of those events. On the back of our successful pilot study, we then established the New Technology Proving Project with IMOS, the Integrated Marine Observing System. And what we set up was sampling across 7 sites Australia-wide. And one of the key initiatives of this particular New Technology Proving Project was to actually focus on the contamination aspect. And so there are quite good protocols in place to consider contamination coming from your laboratory processing and analysis. But at the time, there was not a lot actually looking into how you might control during the field collection component. And so we spent a great deal of time with our field crews to establish quite a stringent protocol in that space. And where possible, they replace plastic gear, where they now adopt airborne controls during the collection. They also have established a vessel-specific contaminant library, which they send to us. And then back in the lab, we then apply our QA/QC protocols to ensure that we do a complete match against the items in the contaminant library with the items we find in the samples. And what this all goes towards is it helps to ensure that we're not actually over-reporting on the problem, because there can be in danger of being, boy who crowed wolf. And so it's really important that we actually can be confident in the accuracy of the microplastic assessments that we're reporting on. So with the new technology proving project finishing in 2023, it was then further funded and is now an established sub-facility within IMOS and funded at this stage to 2027. The footprint of the sampling has been extended to 9 sites, and we've got a 10th site coming on board in early 2025. And what we have been able to do in the last year is really show that there is complete interoperability within this system, within the entire network, noting that we have multiple different field crews that we work with. So I think that's, it was really pleasing to see that we're getting good quality data and reproducible data from the program through these stringent protocols that we apply. In parallel to that, AIMS is also running a regional initiative where, as I said, we're interested in particular in what is entering into the Great Barrier Reef. And so we've applied these methodologies and this strategy to also look at 7 different sites adjacent river mouths from the North Queensland coastline, and we're currently investigating that data. So all the data that I've been talking about, we're collecting that and collating it in a fashion so that it can then be uploaded into the AODN platform that IMOS runs. And the important part here is that this data is publicly available, that a user can go and zoom in on a particular location of interest. They can look at a particular transect if they want to, and pull out the key bits of information associated with either with the microplastic component itself, but also aspects of, for example, the field information, start, stop, the dates, the times, that kind of information. What's really important is that the format that we are using, which is a very simple format, but has many columns within the Excel, within the CSV file, is that this is actually now ready for future upload to other systems such as the Atlas of Ocean Microplastics and NOAA's Marine Microplastic Concentrations platform. So here's a snapshot, if you like, of the data set to date. And I guess the good news from what we're finding is that, in fact, the waters around the Australian coastline are not actually that contaminated. And that's probably to be expected, given we have a relatively low population and we have a relatively high awareness of plastic and plastic pollution. We do note, however, that there are a couple of points that are a little bit higher, and that was the Gulf of St. Vincent and in the orange, and Port Hacking in the red. So as part of data harmonisation, we also spend a great deal of time ensuring that we do a rigorous data characterisation of our particles, and we always report on polymer type, on shape, on size, and on colour. That's fantastic, and the example here is just showing all of the data combined, but we can actually start to pull out our data now because we have over 3 years worth of data, annual data, and we can start to look into what might actually be happening at each site. And so if we remember that Port Hacking was our highest contaminated site, we can now map out this particular site, look at the years, difference in the years, but also difference in the months. And what we're seeing here is, again, we see this massive increase in the concentrations in early 2022. And so again, looking back into a lot of the data, the weather data in particular, and water quality data that's available through IMOS and other systems, what we were able to establish was that, in fact, this coincided with a quite devastating flood in New South Wales at that time. And so I guess what this information is showing, I mean, it seems logical that this would be happening, but I guess what we've now got is actual hardcore evidence, information that we can actually deliver to policymakers and environmental managers so they can actually start to put in place mitigation strategies ahead of these kinds of events. And so over the past 10 years, our plastics monitoring program has undergone quite a massive evolution. But one thing that I would always advise doing is that constantly look and review and update your standard operating procedures. Always look at updating your training modules. We have been continually extending and expanding the program's footprint. And we've actually had a really big focus in the past year on ensuring that our logistics flow work is seamless and streamlined, and also that we're maintaining that really high level of health and safety, which is critical to ensure that all of our field crews stay safe. I guess the main component, however, is, and going back to Amy Lush's comment about, it's really important that we ensure that we have data harmonisation, because as we are slightly changing and tweaking our methods, we still need to be reporting what those changes are so that we can then ensure that our data is truly harmonised and then truly comparable with other studies. I guess one aspect of this also is the stakeholder engagement. And by this, I'm not just meaning going out and talking to policymakers, but also looking back into the system and working back with our field crews, because they need to understand, and with this data in hand now, they can see why it is so important that we maintain such a high level of protocol during the collection phase. And the last aspect I really just touching on, I'm not going to go into all the wording on these this particular slide, but is to say that we need to also be demonstrating that that the data is actually useful, that there is actually an impact from producing this kind of data. And so I've already shown that we can put a spotlight on the problem and that we can develop methods. We're also interested in actually developing methods and understanding the effects and measuring risks of microplastics to marine life. And we're also, alongside our main monitoring programs, are also looking at developing much smaller monitoring programs that are regional for specific clients. And then I guess what's probably really important in terms of ensuring that our data has profile is that we have that we conduct outreach and education. And the big thing we do here at AIMS is we do actually engage with our local community with, for example, the GhostNet initiative is a group of artists. We also have a very strong engagement through the university sector, both locally, nationally and internationally, and also through government sectors as well. And I guess what is really then the next step and is really important aspect of the whole pipeline is consultation with key bodies that are actually going to be the ones using your data to actually try and drive change. And, we've been doing a number of different things with both with government levels, but also most recently we were at Micro 2024 in Lanzarote in Spain, where the plastic pollution treaty is now the main topic of discussion. So it's really fantastic to start to see monitoring being held up as a key component in driving some of these policies forward. And that's it for me. I'd like to thank Marina and Keegan, who are members of the team as well. We're only a very small team, as you can see. But I'd also like to do a bit of a shout out. We have to anybody who's got manuscripts who is sitting around and they're looking to get them published in the monitoring space, and you can align your work with any one of these words in these word bubbles, please contact us. Please submit to our special issue. And thank you very much for listening.
Amazing. Thank you, Sheree. That's really, I'm so jealous of everyone that went to Micro 2024. I feel like everybody's been there. I missed out, but that's all right. All right, so we've got our last presenter now. We're pretty much running on time. We're eating a bit into question time, but that's fine. We've got Dr. Jennifer Provencher, who's, of course, I've already mentioned, come from Canada. At least it's now an hour later, Jen, so it's like, what, 5:30 for you? But anyway, thank you so much for coming. I'll hand over to you now.
Yeah, thanks. It's been exciting to see everyone's work, so I'm happy to jump in as well. I think I'm going to add a voice to many things that have already been said this morning. But I'm going to talk a little bit about how we're working in Canada and how really we're trying to harmonize in the field and standardize in the lab. And so similarly, I'm a government scientist with Environment and Climate Change Canada. And so we work across the kind of macro to micro scale for microplastics. So I'm also the chair of the Arctic Council's Litter and Microplastics Expert Group, and Amy's mentioned these. And we do have a series of kind of guidelines and summaries. And ours came a little bit different than the Australian effort, mostly because our task was to come up with a monitoring plan. But of course, that's easier said than done. And so for those of you who are interested, I'm happy to talk later, but there's the 4-page summary if you want a quick read, all the way down to the 266-page technical version with all the details in it. But really how we see this at the Arctic, Pan-Arctic levels, these are our marching orders for the next few years, right? So we actually did 11 compartments for which you could look for, macro to microplastics. And I'm going to talk a bit about, how we're actually putting this into place and implementing it. And again, the idea was that we were putting together not just guidelines, but a monitoring plan of what we all need to go away and do. So the seabirds is the main focus, and most of you will know that the northern fulmar has been used in the North Sea for monitoring for almost 20-30 years now. This is the kind of region where it started. There's been some excellent work to support northern fulmars, and of course they don't go really small in microplastics for northern fulmars, but we do kind of go down to that 5 millimeter to 1 millimeter size in the microplastics range. In 2020, we actually added this to our Environmental Sustainability Index in Canada. So we have both legislation on the European side and on the Canadian side that use this as a monitor for certain levels of plastic pollution. And again, this is harmonized in both of our sides, and so we can actually do collaborative work. But what does this mean, right? This means that we are doing harmonization in the field. In Europe, they only take wrecked birds that are washed up on the beach. In Canada, we often get our birds from bycatch. We can get them from harvesters, specifically in the north, Inuit harvesters. And sometimes we get them dead on the colony. So in the field, the collections are not the same. They're harmonized though. But the point is that we can go to the lab and share those protocols and actually get standardized data. And so this allows us to do a whole bunch of trend analysis with our European colleagues. And again, I won't go into the patterns here, but we've been able to share data, do joint papers with our European colleagues, and really look at plastic pollution on a North Pacific scale. And again, really quickly, we've been working on this for, at least almost 20 years now in our lab with our European colleagues. And so we can actually see, we get these spatial gradients along a North Atlantic scale. And so we have fairly good data in the Western North Atlantic and the Eastern North Atlantic. And what we're really trying to do in the next little while is really build up our North Pacific data set. And I'll get back to that in a little bit. But you can see that the ecological quality objective, which was set in Europe under the North Sea, in Canada, we fall below that often. In Europe, they're still above it. And so we can actually start to see if those policy targets are achievable anywhere on the planet. And actually, again, have that policy feedback loop to make sure that when we put a policy in place, is it actually going up? Is it going down? What the trend is, and is it achievable? And so there's lots of lessons to be learned in this kind of fulmar work as we again inform that UN Plastics Treaty. The other piece I wanted to touch briefly on was water and sediments. And again, Amy did this a little bit, but again, this is how, this is a monitoring plan that our expert group did in the Pan-Arctic, and water and aquatic sediments are very high. So what does that actually look like in real life is really what we're kind of working on now. So as you are very familiar, many of you are from very large geographic regions, the Canadian Arctic is very big. And so we have been working with a variety of different opportunities, ships, community-based sampling, tourist boats, icebreakers, all kinds of different boats to try to get samples. And so this just gives you a spread of what we've been doing in 2023, 2024. We've got small boats. We've got cruise ships in here, and the different colors are associated with different boat types. What that means, though, is that in the field, we need to basically have a method that can be done by a range of crews and a range of equipment across the board. And so again, this comes down to harmonization versus standardization. So most of the time we make sampling kits. You can see this is a Natasha in my lab shipping off coolers of equipment to some of our Coast Guard vessels and our cruise ships. But other times we have partners, and this is Shelley and Michael and Pond Inlet, where they've had to there's an event and they kind of want to capture, kind of going back to Sherry's point, and they've actually gone to the medical health center and asked for some, sterile urine cups, and they're going to go and do some sediment collections. So again, it's not always the same, but understanding and based on a lot of what our colleagues have done to understand the differences, we can make recommendations and get those samples. And again, sometimes we're on really big boats, sometimes we're on little boats, and sometimes we're working with community groups. And so having that flexibility to employ those methods in the field and understand how they're different is a really important part of our work. But again, and I'll just come back to, and I won't talk about this a lot, but what we've been doing, and Amy talked about this a little bit, is really that standardization in the lab. And so trying to get as close as possible to what everyone else is doing. And I might be a bit of an oddity, but I actually love taking other people's protocols and putting them into work in our lab. I do not need to recreate the wheel. My colleagues often, make fun of me because it's like, no, we don't need to do things. So we actually share protocols with our friends in Norway, and we have a colleague here, Chelsea Rockman at University of Toronto, and we share protocols, and we share in some cases with Chelsea's lab, we share spike recoveries that we can do so that we can do as much as possibly the same. And so again, that gives us the ability to come forward with comparable data. So really the challenge to us, specifically from an Arctic perspective country, is really working with our colleagues, both in the Arctic and outside the Arctic, to really kind of come up with this, I've made this map up, but basically, a pan-Arctic map. So in the Arctic, we're used to having these pan-Arctic maps, for mercury and for lead. We've been doing this for years. It's nothing new. We can't do this for microplastics yet. And so, but our marching orders is how do we do this? And so this is some of the work that we're doing at the Arctic Council to really coordinate amongst all of our partners so we can come up with this pan-Arctic map so that we can start understanding patterns. The other thing I will leave kind of a few words with is that we also, very similar to many people in the colleague, we sit in the Arctic group, we also sit in the North Pacific group. And so I sit on both our Arctic Council group and our Pisces working group for the North Pacific. And so we're really looking at those joint indicators. So again, similar to my colleagues here, in Canada, we've got 3 oceans to consider. And so working across what those 3 oceans look like so that we can compare with our Arctic colleagues, our Atlantic colleagues, and our Pacific colleagues. The way to that end is really just this idea that we're working through with the Arctic. And this was some of the work that we actually just had a microplastics workshop in Italy last week, is really about not starting like, I'm going to monitor microplastics or I'm going to do this methodology. But my students and colleagues will be very aware, but I actually start with mapping your policy endpoint. Because if you are interested in kind of shifting the needle, what you actually need to do is figure out what's your policy endpoint and then design your monitoring to that point. And so I've just put a few examples here, but if the point is to influence the UN treaty, one of the things right now that they're talking about, right, is listing by polymer. Well, that means sources, that means coordinated polymer ID, interlab comparability, and brand on it. And so this is one thing I think that we need to actually also think about, is that we're often excited, we want to go to the field, it's get into the lab, but really what we want to do is that when we're starting to design our monitoring plans, thinking about the endpoint, what we're trying to influence and how do we get there is something that I think we need to do a lot more of it and really think about in a monitoring context. And I'm going to leave it there, and I'll hand it back over to our organizers.
Thanks, Jen. That was awesome. That was perfect. If you could stop sharing, that would be awesome. All right, so Jem was our lucky last presenter, so we're going to open the floor up now for some Q&A. We will have the Q&A open for anyone in the chat that wants to put it in. If you can you put it in the Q&A section? There's something at the bottom that says Q&A. If you can put it in there if you have any questions, I will get to those, or if they need to go in the webinar chat as well, that is fine. I might start with the first question, which is I want to kind of go around the room and we'll go between all the different presenters and kind of share what everybody thinks that the key challenges are that were highlighted during this session regarding harmonization and best practices for microplastic sampling. And if we can keep it kind of like, 1 or 2 sentences worth, so we can kind of get an overview of what everyone thinks. I might just go through. Patrick, do you want to go first, and then we'll kind of go around? Sorry, I'm throwing you under the bus here.
That is fine. I think listening to all the talks and the ideas that came from the work and the work we've been taking on, Nina, and the collaborations we've been having, I think some of the key challenges and ideas to bring in would be that data reporting criteria is a key, and I think we were trying to bring that into our best practices manuals, and having a checklist and making sure that we have criteria that everybody can match and that allows that harmonization. So talking more about harmonization than standardization. And then I think Amy made a really good point also in terms of the multi-matrix monitoring, which I'm talking to the converted when we thought we were doing one matrix and we ended up with 4. And then I also like just to give a third one, this last point that Jennifer brought in about the policy endpoint. So that reinverting the question, I think sometimes the challenge is we want to develop these monitoring programs that are overarching, but probably we might need to think sometimes about what's the actual endpoint you want from the monitoring, and Cherie touched based on that. And that's also a challenge because it's another variable to bring in.
Perfect. Thank you, Patrick. I guess we'll just go down the list. Denise, do you have anything you want to add? Oh no, I think your sound's not quite right.
That's probably better. Yeah,
That's good.
sorry. So I think, to me, one of the things that I often see is we don't design. So it's really coming back to Jennifer's point there that there's a disconnect between the analysis that we need to do and the question that we're actually trying to answer. So I really just want to echo that. Start with your question, know what your output and your outcome desire is, and design from that so that you build in the appropriate approach and analysis in your design in the very first instance. And yes, please don't reinvent the wheel. But I think that's really the fundamental thing, in that we have a lot of people who are really into ecology, who are really into the field, but they may not have the statistical analytical experience to think about design and that importance of experimental design from the front end. And I'll stop there. Thanks.
Perfect. Thanks, Denise. Amy?
I think it's going to be the same as what the others have just said, but most importantly, there are so many activities for harmonization and standardization happening around the world. And I try to keep on top of them myself, but it is incredibly challenging to see where everything is going. And reinventing the wheel is like the central point here. Like, if it's being done other places, we need to come together and work on it together, because some regions might end up doing a lot of work that's already been done by someone else. So it's important to network and understand, or to connect and collaborate globally outside of our regions, because it'll speed up the process. It will help us get there quicker. But most importantly, we need to focus on what that data we're gathering is going to be used for. So back to what Denise says, we need to design things properly, and talking, networking, collaboration will harmonize and hopefully get there slightly quicker. That's my key point.
Perfect. Thank you, Amy. Yeah, I definitely agree with that one. Not reinventing what's already been done. It's saving everyone a lot of time and effort and money as well. That would not, I missed my key point, so I would say that's probably mine. But also I was looking at Jen's map where she had the Arctic and said we want to have sampling all around the Arctic, and I was like, wouldn't it be great to have that all around the world? So you know, we obviously are on other sides of the world, but that would be the dream. Ted, do you want to mention something here?
Yeah, so it's been very interesting hearing about all of the things going on. We don't do a lot of monitoring on my side, but one of the things that I'm curious just to raise is the amount of variation that one gets in sampling microplastic particles in any one location is quite high if you do it in multiple samples and this. And I just wonder how much we're thinking about that. Like, so there's a lot of variation there. Is there as much variation across different methods? So essentially saying how important is it to harmonize methods if the variation is actually really driven by the variation that exists in a site at an individual time? Just a thought.
Can I add a Can I just add to Ted's comment on that? So Ted, I could not agree more, and that is why replication is critical, because variance is so massively important to our understanding, and people look to means and medians, but like we really need to address that, you know, that hypervariability which happens across matrix, across systems, etc. And so to that, you know, like we design everything so that you're actually, for all surface trawl samples, you run multiple short tows so that you can get a mean and standard deviation. And so whether it's trawls, whether it's sediments, whether it's tissues, you know, whatever you're doing, please do replicate so that we can capture that variation, because that is so important for our understanding within different ecosystems and across strata, habitat types, etc. So great point to raise. Thanks.
Yeah, and if I can jump in, I think one of the things that is not done enough is the testing of the methods side by side and the application of statistical framework. So again, I was at a workshop last week where we were talking about this, and our colleagues in Latvia have done an amazing set of work where they actually, for their work, suggest that unless you're doing 3 to 5 replicates in surface water, you're not actually getting enough replicates. But they went from 1 to 20 and then did the statistical analysis of like, what does it mean for them? And so we're doing the same thing with our sediment samples, like how many replicates is enough? Because the answer, you know, for Denise is going to be different than Sherry, and it's going to be different for Amy. And so there's a lot of what I would say, like, and Amy brought this up, but this idea of like testing the methods, testing them again, making sure, and it's not just in the lab, it's the statistical, you know, framing of what are we actually saying and how confident can we say that. And I think there's more and more of that needs to be done. It's not as sexy and groundbreaking. We're not going to get a Nature paper telling people how many replicates of sediment they're going to take, but it's critical to doing good, solid science that will inform policy.
Perfect. Thank you, Jen. Did you want to add anything else while you're speaking about any of the challenges that you think that we are facing to do harmonization?
Yeah, I think I just echo the point that like there's a lot of work going on. And one thing I wanted to raise, again, it's an unpopular opinion at times. It's a critical discussion between what can be done by the public, the general public, what can be done by trained community partners, and what can be done by, professional researchers that are paid in whatever context that in for microplastics, because those 3 groups are very different. And I think we need to be really honest about what those 3 groups can provide along that chain of monitoring. And in my world, we highly advocate for trained community members and what I would call professional researchers. We don't use the general public because of all the cross-contamination QA/QC concerns for microplastics. Macro, meso, different story. We can talk about that. But for microplastics, I think we need to be really critical about what microplastic monitoring tools are good for outreach and discussion platforms, and which are actually going into a data stream. And again, I think that's a discussion point that we need to throw in there.
Yes, for sure. Sherry, I realize I skipped over you, so we'll go back to you. And then I'll go to our next questions. We have a few in the chat as well.
All right, that's fine. Thank you, Nina. Now I'd just like to go back to one of the early learnings that we actually had in our monitoring, which was communication. So, we were working with numerous different field crews. We also, when we started, COVID hit. And so we had this massive challenge about how to actually train people up and how to explain, provide them with detail as to why it's so important that they collect in a certain way. And one of the very first things that happened in our monitoring programs, we got these fantastic samples back, and everything was labeled lovely, and we got all of this data, but the only thing that was missing was the GPS coordinates. So we had no idea of the length of the transects that had been taken for the entire sampling period. And because our protocol required that, we then had to exclude those samples from our data set. And so explaining to people, especially the field crews, what is the importance of what you're doing and why it's so valuable, why the data itself is so valuable. So now our field crews are just like, please let us know, they send us the information and then they double check with us now. So it's really great that you have that strong communication. And from that point of view, I think you have a much smoother ride throughout the rest of the monitoring program.
Great, perfect. I guess that leads on to sort of my next question, which was going to be like, we've identified what the challenges are, and perhaps let's discuss what the key solutions are or how we can sort of address them. And I think what you said there, Shri, is like what I was going to say, which is communication amongst us as researchers, but also with the people that are doing the sampling and people that want to research microplastics, making sure that they're aware of what the best practice is. Does anyone else have anything to add in the sort of solution space?
Yeah, I can jump in. I think one thing that gets lost a little bit in the microplastics world generally is that it's all new. And it is, microplastics is new. But contaminant monitoring has been going on at a global scale for decades. Personally, I'm the steward of a 50-year data set on mercury and DDT and a whole bunch of other things. And so when we're talking about comparability, when we're talking about standard reporting, there's a question in the chat about, do I report in wet weight or dry weight? There are a ton of lessons to be learned from that kind of base contaminants literature. And there's not going to be, one solution to it all. There's not going to be a golden, answer. And that's okay. But I think we can learn from the contaminants community who has influenced the Stockholm Convention, who has influenced the Minamata Convention, ozone layer, all kinds of things, and really take those lessons and build them into our microplastics program. So we feel like we're starting from scratch, but we're actually not, and there's lessons to be learned. So I think that's a solution, right? Reaching across to colleagues either who also work on contaminants like myself, or who have worked on contaminants, but maybe not microplastics, and talking to them about how did they set up a global monitoring system? How did they do all these things is a real source of knowledge and experience that we can tap into.
Perfect. Anyone else have anything to add in this solution space? Otherwise, Patrick?
I was just going to say one of the interesting points that's been raised today, in addition here for the replication and the power analysis and bringing in the importance of survey design before we start and collect. Another solution of interest potentially is thinking, can we enhance, I think it was Jen that mentioned that had a spiking recovery program with other universities. Can we enhance that collaboration and start bringing those more in across our labs or several labs in a way to promote intercalibration of data sets and tap into previous data sets that we may have, and potentially, this is a bit of utopic, can we go back in time and join if we find how well we can intercalibrate, but it's more more on that, on the spike in recoveries and identifying that we're actually doing the same.
Yeah, perfect. All right. No one else to pop in there. Otherwise, I've got one more question before I'll go to the Q&A. This is a question actually that I had for Denise, and also Amy and Ted and Jen probably can all pitch in where they want. But how do you think the challenges from sort of a Eastern Asia perspective, or from any other resource limited regions, how do you think they compare to places in Northern Europe, and I guess the sampling that we can do between those sort of two regions? Your headphone again. Wait, your microphone.
Well, yeah. So let's broaden that out a little bit, right? Because I think it's a falsehood as well to focus and to think that so much of what we find in the environment is coming from Southeast Asia, which has been really highlighted. You know, we're failing to consider Africa and other areas as well. There are a lot more resources in more economically advantaged countries, and there is a corresponding increase in labor cost in many of those countries. So while you may not have the same financial resources often in more financially disadvantaged countries, what you do have is a broad coalition of the willing, which in the microplastic space talks to the ability to do some sampling in certain areas, and also to be mindful of, you know, the point that Jennifer raised about ensuring that people that are working in areas are trained. And so, you know, I think a lot about capacity. We do a lot of capacity building. I'm involved in a couple of things. One of the comments in the session in the chat box is about, you know, the IAEI methods development, and both Amy and I have been involved in that, you know, which is a real opportunity to broaden out some of these protocols, etc., into a suite of different countries of geographies all around the world. You know, I think there's a real necessity to, and we do see that in some instances, there are actually resources being made available to countries through the UN for some of the expensive lab equipment that's required to actually undertake some of the, you know, more advanced microplastics sampling and that sort of thing. So, you know, I think, you know, there are universities that are focused on microplastics, it seems, in nearly every coastal country around the world. So I think there's really good opportunity and collaboration. I would also touch back to that recommendation of not having so many overlapping groups developing different methods and handbooks and protocols, etc. Because we do see that over and over, you know, and even in Southeast Asia, there's, you know, at least 5 overlapping organizations that may involve some but not all of the different countries within even Southeast Asia alone. And so, you know, making sure that we're having that really good communication that's been highlighted as well, I think becomes really important. Does that address their question?
Yeah, that answered the question. Yeah, I think Sarah had a question that kind of aligned with that, I guess, on the chat, where she was saying she was interested in the thoughts on approaching lessons learned in terms of harmonization with researchers and groups from less affluent countries, areas in the world than the countries represented on the panel. And I think that answered it quite well. But if anyone has anything else they wanted to add to that comment, Sri?
Yeah. And the other thing I'd say is most of my work isn't even in Australia. So I do, most of my work isn't domestic. So we do reach across those areas. Sorry, back over to you, Sheree.
No, you're right. You're right. No, I was just going to say, so we don't do a lot of work in the Southeast Asia, but we do have some strong collaborations with other countries, including Brazil. And what we've actually found is we've actually been engaging in a program with a couple of universities whereby they come to our lab. We train them up on the methodologies, on the thinking behind the methods that they're using. teaching them the techniques using our equipment, and then basically arming them so that when they go back home, they can go back into their institutes and say, right, this is why we need to focus on putting as many resources as we can into training people in these areas or into getting particular equipment. And we're really stoked just recently that one of our students that came and stayed with us for 12 months actually went back to her home university, and one of the vendors actually said, oh, well, we'll give you an instrument because we can see that you're establishing a new group and a new area of research that is not really something that Brazil is taking forward, at that level of rigor. So I think that there are other ways to do it rather than also going into the communities, but also bringing them into our labs and doing the training and sort of building up their expertise. But that's been our approach to it.
Awesome. Does anyone else have anything to add in that space? I'll move on to the next questions. All right, cool. So we have a question here from Sakthina Rendaran, who's a research scholar from India. And they're asking, is there any standardized protocol for assessing the acute and chronic toxicity of microplastic and co-contaminants? I'm kind of thinking of this, you mean in an eco-toxicological way. I'm not entirely sure on the phrase of that. Does anyone have anything they want to add there?
Well, I guess I'm happy to sort of respond to that we're very short on standardized methods for testing those materials like that. We've been working on that in for quite some time. The topic of the co-contaminants or associated substance equally is without really standardized methods for doing it, and that's one of the challenges that exists.
I'm going to jump in. So in North America, we have standards for, we have certain species, we have to say, lethal dose 50. Like there are some standard, I wouldn't say methods, but like endpoints that people really understand and can put into their risk assessment work. The challenge that we're finding in our group is that the standard species and the standard metrics or endpoints don't necessarily fit a microplastics concept or real life situation. For example, there is a bunch of zooplankton that all of my water colleagues use. They've got standard tests. They, measure the lethal dose. That's what goes into our risk assessments. But what we're finding in, this is freshwater, so it's a little different, but what we're finding is that we have a lake experiment where we've dosed it with plastics. 99.9% of the plastics, regardless of polymer type, are at the bottom. And so there's this difference between where soluble chemical contaminants are in the water column that we use for toxicity testing, and then where the plastic is actually at the surface. Most of it's actually at the bottom. And so there's a disconnect between our standard toxicology methods, at least in North America, and where the plastics is actually found in the environment. And so I think that's part of it. So I think there's two pieces to that. Are there standard endpoints and standard toxicity tests? Absolutely. Are they applicable for microplastics and co-contaminants? I don't think they are. And so we have to be, again, critical on like what pieces can we borrow from toxicology and what pieces do we really have to think about as new because of the specificities of plastic pollution in general.
Okay, that is a great response. Does anyone have anything else to add to that? Sorry, I'm trying to monitor this chat and it's just confusing me because I don't know what button to press. Does anyone have anything else?
I was going to come back to that. Nice points there, Jennifer. Yeah, absolutely. There are certainly standardized tests for doing toxicology. The issue, I think, with the plastic particles and other particles is that the trick is getting exposure to be reproducible, and it really makes the challenge of getting any repeatable data, even within the same laboratory, a real difficulty. And then likewise, I think with the co-contaminant topic, that's adds even a greater complexity to it.
All right, that's perfect. We'll move on to the next question now. So Keshni said, great presentations. Thank you, too kind. UN, IAEA are working on harmonising microplastic techniques. Currently, there are over 61 member states involved. The aim is to provide comparable results for SDG reporting. Most of the countries here, not UK, are not represented at this. Is there a plan to be involved as you are all member states? Denise, you mentioned something on that before, and I did hear you say something about something and then maybe pop in here.
Oh, so Amy and I have both been involved in one of the earlier rounds of the development for these harmonization of microplastics measurement and monitoring. Amy, you may want to chime in more. We were both meant to be involved with kind of the development, the technical development, to provide that input as science experts for that. Countries need to invite people to participate, or you come in through another process. But I understand the next meeting on that, because Amy and I were chatting in the side, where that will be coming up as well. So I think some of us at least have been involved in such international efforts and working groups. Amy?
Provide a little bit of extra context, maybe, for those that aren't familiar. So the IAEA has been scaling up this harmonization of microplastic techniques. It started in Latin America, and then it was scaled to the ASEAN region, and now it's being scaled up to Europe, I believe, and then internationally after that. So the purpose is the methods are developed primarily within Latin America and South America, and those methods were then being transferred to each individual country. So the idea is that the methods will remain the same. The individual countries are, theoretically, every country is going to end up being involved in this, and it's a representative of each country. Is not necessarily a monitoring organization. They are more closely associated with the what's the word I need? The Nuclear Technology Institute. So it's usually somebody put forward from the link to the nuclear National Nuclear Laboratories. I can't remember the precise wording there. So yes, it's scaling up, but it might not be linking to those that are currently leading microplastic monitoring efforts in individual countries. So I know there is efforts to align the IAEA ambition alongside the also national and regional ambitions that happen to be going in parallel. So there are efforts to try and align that to make sure it comes on board. But I think that's the best I can say about it at the moment.
And
I'm gonna jump in. Can I just jump in and talk about, please. I think the importance is that there's a lot of groups and there's a lot of researchers, but the importance is networking, and we can't be everywhere. So I think just recognizing that, I talked to Amy, if Amy's on a committee, I'm like, oh, great, she's got that one, right? I can go to do this other one. Like, we can't all be everywhere, but that's why dialogue and networking and having sessions and having projects together are so important, because we can't do everything. And so I will admit there are times when I see a committee or see a meeting and I look at who's already on it and I'm like, nope, they got this. It's okay. And then there's other times that I'm like, ooh, we could bring something to the table that's not represented. So I think there's also that piece that, like, we can't be everywhere all the time, but the reason why we don't have to be everywhere is because of understanding and collaboration and constant networking and dialogue. And that is so critical to a global effort.
That is an awesome comment. I think hopefully this workshop in its own way will also help with that, contribute to that, because I have never connected with Amy or Jen before. Now we know each other, so it's a good way to make sure we keep I guess the ball rolling. There's a couple more questions that I just want to quickly get to because we're running very short of time. This might be another question for you, Jen, because we're not in the up north in the Pacific garbage patch, but there's a question here that says, are there any monitoring works in or near Pacific garbage patch?
Yeah, I wouldn't call it monitoring. Monitoring, is like repeated all the time, but we certainly are doing work. So most of the work in the garbage patch is through bilateral effort. So we actually have a boat where Canada and Japan have done a joint mission. So we've got boats going back and forth. and they're using harmonized methods that we've put in, and we're going to produce standardized data together with Japan. So the answer is yes and no. Because it's international waters, that kind of responsibility for monitoring is a little gray. But I would say, again, through Pisces, which is our North Pacific, regional group, we have done quite a bit of work to try to bring that together and to promote bilateral. So the answer is it's complicated. I wouldn't call it monitoring, but I would say that we are working on many facets to get more data so that we can move towards answering those standardized questions.
All right, awesome. Okay, the next question is from someone saying, is there a standard measure for microplastic abundance in sediments, e.g. particles gram dry weight, particles per kilo dry weight, particles per gram wet weight? I think the answer to that, I could comment on that saying, I think all okay, you just have to report, well, ideally you report them all and report your raw data so people can calculate that if possible. Does anyone have, everyone nodding? Yeah, good, I nailed it. And then also the preferred reference library for microplastic spectra. I don't think there's a particular preferred one. There's some that are free, some that are not. Again, reporting on it, saying what you use is key. Are there any that people love that they use, if anyone's like wanting to highlight one?
Well, I would just say that there are some that are open source, and there are some, I think the bottom line would, you have to have something that includes weathered polymers. At least from my perspective. So I wouldn't say that there's like a preferred library, but I would say that there's a bit of a checklist to go through to make sure that it includes, and from there are lots of options, including some open access ones.
Awesome. No other comments on that. Wow, ticking these off. Sarah came back in and said, can you just repeat the organization that was mentioned that supports funding? It sounded something like AEI. Was that something that Cherie said about the funding? I don't know if it was that program you were talking about, Cherie, or if it was talking about the UN IAEA. There's too many acronyms going around here. I don't know if we know the answer to that one. Oh, sorry.
It wasn't from me, so sorry.
Okay. It might be that one we said at the top. Hey, I think that's all the questions we had. We had a lovely comment from a few people in the chat saying, oh, Sarah said she found it. That's good. Saying how helpful it was, and Marina had a great comment talking about the great point that Jen made on regarding learning from other contaminants for monitoring, and then also a bit more about I'll let anyone read that comment because I think everyone can see it. Marina, great comment though. I think if no one else has any burning questions, we might tie off because we've gone a little bit over time than what I planned. I guess we did have one more kind of broad, thought process, which was how can we harness this momentum that we have and use the Ocean Best Practice platform going forward. And this is an Ocean Best Practice platform that we're using now. So I think just continue networking. It sounds like there's already a great network of people going on, but I know from an Australian perspective for us, like making that more global is really important. Anyone have any comments in that space, or just?
I'll just say that, so I had the really good fortune of going to Micro 2024 this year, and I've not actually been to a European-based microplastics conference or forum where plastics have been mentioned. And I have to say, it blew my mind. It was amazing. And all I can say is get out as much as possible. I know COVID sort of, stifled that interaction a bit, but I was so impressed with what I saw over there and with what the European, and there was a couple of American groups and what people are doing in that part of the world. And I just so wish that we were a bit closer. So yeah, full engagement, get out there, network. It was great.
Well, that's awesome advice. Yeah, I did see everybody. I got these out of offices at the same time and I'm at Micro 2024. I'm not at Micro 2024. Is there going to be, when's the next Micro?
Well, I don't know that it's actually been announced, but I think it's 2 years' time.
Yeah, it's usually every 2 years, I guess. Just keep an eye out, and there'll be some developments from I'm sure the steering committee. They usually get in contact a year in advance, so just keep your eyes open.
Okay, Micro 2026 after. I think my boss is still on the call. That's a good highlight. Okay, cool. So I guess we'll close off here. I just wanted to say thank you so much to everyone, the presenters particularly, for coming. All of your presentations were so, it was amazing to see the synergies, but also how they're unique in their own way and get that global picture. There are, of course, areas that we've missed, and you can't do everything. As Jen was saying earlier, you can't do everything, but I think hopefully I've done a bit of a broad overview for those people that are new in the space, or at least for us also to network and collaborate for some next steps. It's amazing to see like such a passionate group of researchers, which I think seems to be a bit of a thing in the microplastic space. Everyone's very passionate, which I love. For those people that like want to pass this on to friends or colleagues, etc., there should be a recording on YouTube. So feel free to pass it on. Otherwise, my email is always open, and I think most people probably shared their emails as well. I think that will be it from me. So great work. Jen, go back to bed. It's probably coffee time now.
Well, the sun's almost up now.
Yeah, now you're awake. It's too wrong timing. But otherwise, hopefully we'll catch up at some point in the future. And thanks to everyone for coming.
Thanks, Al.
Thanks heaps, Nina. Thanks, everyone. Bye.
Bye.
Thanks, everyone. Bye.